Welcome to our webinar, everyone. Thank you for joining us today. So today, Víctor Medina will be speaking. Víctor's from Probiomed, and he's going to be speaking about. So Víctor published a paper with us in 2017, and this is on the determination of potency of TNF, anti-TNF alpha antibody, monoclonal antibody. So he's going to be giving us some updates on the procedure that's, kind of happened over the last couple years. All right. So, please, if you have any questions, you can answer those in the Q&A box, and we'll address those towards the end. And I'll also say that we are recording the webinar, so we'll send all the attendees and registrants a video file of the recording afterwards for your records. Okay, Víctor, I will leave it to you.
Well, thank you very much, Ronald, for your introduction. Good morning. Today we're going to talk about a biological bioassay developed to test the ability of a mAb to neutralize TNF-alpha cytokine that is related to arthritis or some other disease. This neutralization assay was developed in order to report to the health authorities in our country. So let's begin. Well, we're going to give a little introduction, then we're going to speak the main bottlenecks that we have and the solutions that we get in that time to the development of this method and its validation. So, and after with all this information acquired during the development of this method, we developed some other biologicals. So we're going to talk about a case study. So the biologics are, in order to understand where we are going to be in the large bioprocess assembly, we can follow an easy diagram where the function of a biologic is determined by its structure. This structure gives the physicochemical and biological properties of biologics, of mAbs, and these properties gives them some kind of safety and efficacy. These mAbs, well, these bioassays could be separated in different kind of bioassays that could be subcellular organelle-based bioassays, cell-based, cell or tissue ex vivo, called ex vivo, with preclinical studies and clinical trials. Within the in vitro bioassay cell-based, we can have a lot of them. We developed some of them for mAbs, others for EPO, others for interferons. And we developed these methods in order to report to the health authority in our country. So between the most important or kind of bioassays are the enzymatic, antioxidant, anti-cancer, antibacterial, and today we're going to talk on neutralization 1. So our method consists in the principle of this analytical method consists in the detection of luminescence generated by the hydrolysis of luciferin covalently attached to a peptide. The hydrolyzed luciferin is liberated into the wall and processed by luciferate, liberating light, and this could be detected by a spectrophotometer. This spectrophotometer must have a luminescence function, so it could be detected very easily. So the main parts of this method are the preparation of media, solutions, equipment, cell culturing and counting, antibody preparation, neutralization assay, and then the analysis of data. However, we are going to talk about troubleshooting mainly. These problems that we have when we were validating and developing this analytical method. So the availability is very important. Why? Because if you have unhealthy cells, you're going to have a lot of trouble with the baseline obtained the method. So cell viability is affected by different factors: the composition of media, that usually is provided by the spread vendor. And this information is very useful because they have the strain. So they have experience cultivating that strain. I think that the most important component is the fetal bovine serum. FBS finally concentration can switch a healthy and successful culture to a stressed one with a noisy signal in your biological assay due to the death of the cells. So this debris is very important to maintain very low. So to get also the culture conditions of your assay is very important. Temperature must be double checked with analogic thermometer, because sometimes your equipment could be malfunction due to the change in electrical supply. So you must to check, at least double check temperature, also the carbon dioxide concentration, and also the physical chemical stress that you're going to give to your cells is also very important. Finally, the cell viability count using equipment that used to count and it's automatic would be very useful to get good results. I remember that when we were developing this method, low viability count give us very noisy baseline. So you must to check that very well. Then also cell mixing is very important. So you must to mix very soft and constant, about 3 or 5 times pipetting before dispensing samples into the cell counter, because you need to have the same amount of cells each well. So bad mixing of cells and different number of cells in the plate is going to be catastrophic to your analytical method. So you must have to take a look to the way we have a mixing. Iniciar mi video. Here is it. You can see it? Yes. Excellent. So the way you are going to mix is very important, and that's critical. Also, another problem that we had when we were developing this method was the stress, the cell stress. The TNF-alpha by itself can't kill cells, so you must to give them another stress signal. This stress signal, we have a lot of trouble at the beginning, because we wanted to use actinosin D, that is an antibiotic that intercalates into DNA. So sometimes the cells were killed completely, and some other times the cell were not killed when the actinosin was when the TNF-alpha was neutralized by the mAb itself. So we thought in that time that FBS concentration, well, FBS is very important for the cells. So we reduce the amount of FBS in the assay from 10% to 5%. And so we get better results. So at the end, we finally take it to 1%, getting very good results. So this stress signal was a real success after some months of investigation, of research. So the other problem that we had was the cell signaling. So the problem was at the beginning, we begin with MTT, but the problem is that we read at the spectrophotometer absorbance, but absorbance cannot be separated from light scattering. So if you have a lot of cells that, and also the cells by themselves gives you absorbance. So there's a lot of noise signal when you are using this method. So even if there are a lot of papers that talks about MTT use for cell viability testing, we were not able to get good results with reproducibility of the samples. So I think that for beginners, it's better to use Alamar Blue, the Resalsurin, that is a fluorometry method. This method, well, the Resalsurin can penetrate to the cell membrane and can be reduced, so it can get a fluorescent metabolite called resorufin. So this method gives us very good results, and also it's cheaper than the other kit while we're using luciferin. So with all this knowledge that we get from this paper, we wanted to explore some other biological methods, and we could publish this paper, and it's about the function of glycosylation in etanercept. Etanercept is a molecule that has two main domains, the Fc, and is a fusion protein, where the Fc of a mAb is fused to the TNFR domain. So the TNFR domain can join or interact with TNF-alpha, inhibiting its function in vivo, also in humans. This is a biological that is right now selling the market. But however, it doesn't have a CDC. CDC is a complement-dependent cytotoxicity. So in the Fc part, it can interact the C1q molecule. So, but however, in this molecule, it's inhibiting this biological function. So we wanted, well, the hypothesis was that glycans, and this structure is highly glycosylated, can inhibit this process. So we have a model in which our biological assay was tested, where the CHO cells that have TNF-alpha expression on its surface, and it doesn't have the enzyme that cleaves this TNF-alpha to the blood or the media, so it can interact with etanercept. However, as C1q can't interact with etanercept, so the cell is going to survive. So what we have done, we have cut one by one all these glycans. First, well, we cut the N-glycosylation. We can cut also sialic acids, galactose, and finally all glycans. So what we have with the intact molecule, when you get to the right, you have lower potency. And when you get to the left, you have, lower potency, higher potency. So the intact molecule does not have CDC activity. Also, when we deglycosylate in the N glycans, we also do not have. Then when we cut the sialic acids that are all negative charge, highly charged, we can get about 10-fold activity of CDC. So with all this knowledge get from the biological assay, it's led us to understand how, for example, etanercept can have different biological functions. Well, I think that's all for today. If you want to ask a lot of more stuff about this, I'm open to this. Also, you have there my email, so you can ask me personally if you have another question. and this is our team in our lab. So everybody has a part in the development of this method and some others that we are not right now doing. Also, we are doing some other biologics right now. Well, a lot.
All right, thank you for the great presentation, Victor. It doesn't look like we have any questions. I think, Victor, you gave a very clear presentation, so everyone is very clear with your methodology. So yeah, we can conclude here. So thank you everyone for coming, and thank you, Victor, for the great presentation. We will
Thank you
send
very
a
much for all.
Yep.
Yes, thank you very much. So see you.
Take care, everyone.
Ciao.